Okay, students. Am I audible or
Not audible properly.
okay? Okay. Is it fine now or not? Actually, the network is very low.
Yes, ma'am, it's fine.
I'm using the Amity Wi-Fi. It could be right. Okay, so I guess most of us are here. Only few are left. So it's okay. We'll anyhow start the class. So
Ma'am, excuse me.
yes.
Ma'am, I think we can ask now
Ah,
about the.
yeah. Okay, students. So I was lately I was asking Devanshi about the practical midterm exam, right? Plant pathology. So I mean, I suggested, okay, if you all can get this Friday, we can publish one extra class, and or just your opinion. I mean, when you all want to give. We don't have much time though, right? So today is 14th. We just have this week and next week. That's it, right?
Ma'am, is it possible we can do on Wednesday?
Next Wednesday? Yeah, no problem. But
Ma'am, this Wednesday.
this? No. No, not this this Wednesday. No, I'm going to take this exam offline, and so because I want to take Viva also. So let's do it when you all are here, right? Not. Mm, I mean, enough of the online mode. Let's do something offline. Okay. So let me see. We have this whole this Friday. Then next, do we have class? Just a moment. Let me open the Amazon also when we have the classes, right? Okay, so we have class on uh, Tuesday also, right? Tuesday we have that. So we can put one extra. We can do Tuesday. Then we can do next Wednesday is little packed for me for other classes also. Or Thursday I don't want to take because Thursday we are already doing the PPT part, so we'll lag behind there, right? Or maybe February. 25th February is also fine for me. Next, so it's up to you. You guys decide when you want to give. Just let me know, and it will be all fine. Practical, theory, other theories we have already done, so it's fine. One exam of time is here in it, right? So you guys can think about it and let me know because आपके और exams भी होंगे, so that's why I want to fix the date early because then there will be a lot of traffic and lot of things and you'll be complaining that you are already giving a lot of exams. So इस Friday अगर आप दे देते हो it's best. I mean you'll be free, I'll be
Ma'am,
free.
we are okay with 25th.
25th year okay fine then book the date and please I don't want to hear then कि मैम हमारा कोई और एग्जाम है we cannot give या फिर something like that. To book the 25th date for our plant pathology practical, no problem, right? Okay. Okay, so let's start with the today's topic. Today, I will discuss a very small but a very important topic, which is about RNA interference or the gene silencing, right? And some application of RNA interference or the gene silencing in the crop improvement, right? Which is so. To start with the RNAi technology, or it's a similar term, right? We sometimes we call RNA interference and we call the gene silencing, or we call sometimes PTPS also, or this phenomena is called co-suppression or calling also, right? And this important phenomena was basically discovered by Fire and Mello, and they got a Nobel Prize in 2006 for in physiology or medicine for this. So they worked on nematode and they published this finding in around 1998 around, right? So they explained this phenomena, and then later in plants also this phenomena was being studied by, I guess he was Nepoli et al., some other worker, and he published his findings about the plants, that how in plants it is being done. So again, it is again, we talk about something, when we say that gene silencing, we say that it is a post-transcriptional gene silencing or it is a transcriptional gene silencing. Usually, both, the pathway is similar in both the cases, but when we talk about transcriptional gene silencing, so there's a promoter methylation, right? When we talk about the post-transcriptional gene silencing, so there it involves somewhere the sequence-specific RNA degradation. The basic mechanism same here, but yes, the terms that we use are different, right? Then there are siRNAs or there are microRNAs which are being produced, and both are very important in RNA interference work, right? So yes, these two also differ, siRNAs, when we talk about siRNAs or when we talk about miRNAs. So they too also differ in the way of origin, because siRNA, they are exogenous double-stranded RNA which are taken up by cells, right? Whereas siRNAs, they are also present in lower animals and plants, but they are not found in mammals. Whereas microRNAs, they are found both in animals and plants, right? So there's little difference between them, but we are not going into the real mechanism. What we want to see is that why PTGS or gene silencing mechanism is useful, and how it has been discovered, and what are different research examples of different research where this mechanism has been used in the improvement of the plant, right? So that basically we are going to discuss. So when we say RNA interference or transcriptional gene silencing or virus induced gene silencing and these microRNAs, there are a lot of mechanisms where these actually lead to suppression of a gene expression in the plant, right? So basically they suppress the gene expression, certain genes. These mechanisms, they again, there are a lot of biochemical pathways, right? Dicer enzymes होते हैं, risk complex होते हैं, how they bind, how the dicer enzymes swap the double stranded RNA into single stranded, and कैसे वो silencing करते हैं. It's a different mechanism, right? But this powerful biotechnological tool has provided plant breeders and researchers with a good opportunity to accelerate breeding programs. This Since this I've taken from a paper which was talking about the woody plant. So here we are talking about the woody plant, but otherwise in other plants also this mechanism has been very commonly used. So gene silencing is usually when we say about that how it is defined, it's a process or a molecular process which is involved in the down regulation of a specific gene, right? And probably it has evolved as a genetic defense system against the viruses and evading nucleic acid. So that's why in plants we always think that PTGS is a phenomena which is being used by the plants against the viral pathogens which are infecting them, right? So currently there are several groups of gene silencing which are identified in the plants, such as PTGS, post-transcriptional gene silencing, or the RNA. Just am I audible now? I guess there was a network glitch here. Can you see the PPT now?
Yes, ma'am.
Okay, fine. So again, all these pathways, they play important role at the cellular level. They affect differentiation, gene regulation, and protection against viruses and the transposomes. So there are numerous possible applications of a gene silencing in the plant. So initially, when this was identified in the early time, so main research focus was on the production of a virus-resistant plant through genetic transformation using this technology. Again, this technique has been used in the food quality modification, such as reduction in the caffeine level in the coffee beans, or maybe to increase the nutritional value of a corn, proteins, and tomatoes. And this has also been used in a research on forestry yield and quality, has included the study of a gene silencing related to the lignin synthesis. So in number of ways, according to the type of the plant that we are using, it can lead Oh, I'm sorry, I'm getting disconnected. I'm connecting again with my mobile. Just let me do it. Yeah, yeah. Am I audible now?
Yes, ma'am.
Yes, ma'am.
Okay, fine, great. So there are numerous applications of this gene silencing in the plant. So initially, what they did, they started with the production of a virus-resistant plant by using this mechanism, and then again, this has been used in the improvement of the food quality or modification in certain particular, such as they reduce the caffeine level in a coffee beans, maybe probably by down regulating the genes which express the production of these caffeine compound in the coffee beans, or they were able to also increase the nutrition values of certain plants, right? So its use basically depend upon it was a diverse when you what we exactly want to do, whether we want to go for a resistant plant production, whether we are looking forward towards the improvement of the crop, right? Crop improvement. So it was diversely used in many ways. On the other hand, research on the food crops has again targeted application of a gene silencing on viral and bacterial resistance, right? Then study of plant gene function by affecting gene expression through gene silencing techniques has also been present in the recent line investigation. So it has number of applications where people were trying to use this technique for a variety of the they use it or in whatever different ways they can use, right? So this phenomena was known as a co-suppression also because when okay, this I missed something, right? Yes. So on the fruit trees, it was done. Then the study of plant gene function by affecting expression through gene silencing or PTCS or virus-induced gene silencing. This was also done. Then some of the yeah, this is a great story. How it was being identified in the plant. So from Napoli was the person who identified it in the plant. So when he was trying to obtain transgenic petunias. So by using the greater amount of anthocyanin pigments, right, by amplifying the gene activity of chalcone synthesis, which is involved in this. So what he was expecting that he will obtain a deeper purple color petals, right? That was what he was expecting while he was doing this transgenic experiment. But instead, white or chimeric flowers were produced. So then he thought that why it happened, why, I mean, while he was trying to express his gene in the plant, why it was not expressed. So it apparently he found that the transgene was not expressed and it ended up silencing the homolog endogenous gene, right? So this phenomena was named as a co-suppression and it was unstably transmitted within a generation, leading to the hypothesis again. So this was not for a single generation. This was taken up to the other generations also, which leads to the hypothesis that it was mediated by nucleic acid or the RNA. There was some role of a nucleic acid here, right? Then similar phenomena were named swelling in fungi, RNA interference in the nematodes. These research indicated that presence of a double-stranded RNA or dsRNA, a non-occurring form in a normal cell, was related to the silencing of sequence which were homologous genes, which were similar, right? So in fact, the phenomena it seems to be triggered by the presence of double-stranded RNA suggested that this could be originally a defense mechanism against viruses in the plant, right? Since they found that this phenomena was more found in those pathogens or viruses which were ever standing and they were infecting the particular plant. Then most of this phenomena is related to the RNA activity within the cell. So therefore, we use the term RNA silencing um, as more often as compared to the gene silencing, right? And it combine all the mechanism by which RNA sequencing they regulate the gene expression, right? So again, the study of similar phenomena were done in another organisms also, like in a nematodes, it was done in Neurospheric it was done, then it was done in the Rhabdopsis thaliana also, right? So different models were proposed for different for the underlying mechanism against the gene silencing, right? So Then applications of a gene silencing in a woody plant. Since this paper is being taken is about a woody plant, so we'll be discussing that. But in general, application of a gene silencing. Yield of a woody plant is a result of a growth that depends on the several aspects of a physiological machinery. Now, gene silencing is a very powerful tool here. It can lead us to the better understanding the organization of this machinery, especially the biochemical pathways and its function. Now, woody plants have an economic importance, in agriculture, right? Wood, cellulose, fruit, resins, rubber, oils, they all are derived from the woody plant. So, in order to guarantee the sustainability of woody crops, it is important to keep the high quality and the yield levels. For this purpose, conventional breeding, we know we all dependent on the conventional breeding programs at one level. But these programs, they were not enough to supply short-term solutions for these crops because it takes a lot of time. So because woody plants, they have a slower growth than the herbaceous plants and they live longer juvenile periods, right? So these characteristics speed down the breeding projects in this area. So similar problems occur in the woody fruit crops breeding as well as others such as polyploidy or the pollen and ovule sterility or sexual incompatibility, right? Where the limited progress towards the production of a new cultivar through conventional breeding program was available or it was not possible. So after the genetic transformation of the first model plant tobacco as well as other, so then they started to work on the developing of the transgenic woody plants also. Now this has proven to be a challenge since woody plants they are not very easy to transform and they require longer evaluation process. More than 16 species of woody plants were transformed between 1988 and 2000. Now, these transformations were focused on the traits such as herbicide resistance, insect resistance, virus resistance, lignin biosynthesis, marker genes, and self-therapy suppression. Some of these fruits are obtained by specific transgene expression into the proteins. However, others were achieved by silencing of transgenes or the endogenous gene. So they used these particular traits to improve the yield of the crop or maybe the quality of the wood of the crop. Then gene silencing when a plant disease resistance. Woody crops are particularly susceptible to systemic viral diseases because of their long-term span. Lot of viruses are being identified on the path. Once infected by viruses, we cannot do, as I told you earlier also, the viruses cannot be managed if they are in the field. So we don't have any option, we just need to destroy the plant or destroy the orchard if we want a healthy, we have to start from a scratch, new plant, new trees, right? So virus disease resistance in plants may be achieved again via CP4 protein mediated protection or antisense RNA, right, or PTPS or RNAi or pathogen derived resistance. So there are a number of concepts. So pathogen derived resistance suggests that the resistance to determined pathogen could be obtained from its own genetic material. So इसमें क्या करते हैं PDR में usually हम क्या करते हैं we use any gene of the pathogen. For example, if I want to derive a pathogen, I want to develop a virus resistant plant to the cucumber mosaic virus CMV. So what I'll do, I'll use some of its genes. उसी का किसी gene को maybe coat protein or maybe two these genes, human protein genes. I'll use that gene, I'll introduce into the plant, and that plant will be eventually resistant to that particular pathogen. So that resistance is called as a pathogen derived resistance. So we are using pathogen genes only to confer the resistance to the plant, right? Then quest for virus resistant plants. Again, this was PTGS was also a phenomenon which was accidentally discovered, and again this is again being used as a major strategy in plant transformation for virus resistance. This is a CPV, citrus. This is a virus resistant citrus plants which express the core protein of virus alpinectin. So by introducing the core protein gene into the plant, they have developed the plants which are resistant to this particular virus, right? So how this works, how the pathogen derived resistance works. So the hypothesis for this was also the answer to this was also the RNA silencing or the PTPS silencing kind of a phenomena which are taking place, right? So despite the potential application of GS as a viral defense system, there is a drawback Yes, this is interesting, right? So viruses have now developed RNA suppression strategies during coexistence of the plants, right? Evolved तो हम सब बोल रहे हैं, right? If plant is evolving via PTPS, then viruses are also evolving. They need to come up with the phenomena where they can suppress this gene silencing, right? So this these days now RNA and DNA viruses they have certain silencing suppression proteins in their genome, right? Incorporation of a certain silencing proteins into their genomes, which silence these kind of a gene silencing or PTPS phenomena, right? So, um, okay. So in plant viruses, number of proteins are being identified which act as a silencing of the suppression or a gene silencing, right? For example, 2B protein in cucumoviruses or maybe helper component or the movement protein in certain viruses they are being identified that they can act as a suppressor of the gene silencing, right? So this with this way the viruses are also evolving, right? So gene silencing and wood quality, food quality and other traits. So during chemical pulping of wood, one of the most expensive and hazardous processes to separate lignin from the cellulose and hemicellulose, right? So the production of plant material with lower content of lignin would mean significant reduction of the cost and pollution to the paper industry. So one of the approaches to obtain reduced lignin forest trees has been down regulation of the lignin biosynthetic pathway. So they can just down regulate. They just try to down regulate those genes which lead to the production of more lignin, right? So the main genes which are involved in a genetic transformation targeting the lignin are 4-coordinate or the coenzyme A ligase number of genes which are involved, right? So down regulation of these genes can lead to the reduction in the lignin content, right? And which is again compensated by increase in the cellulose content. So this is how they are useful in the trees and how a down regulation of a certain gene can be helpful, right? Then silencing was also registered when trying to boost up expression of static acid in populus alpha, right? So this अगर ये हम पे डिपेंड करता है अगर हम एक इस जीन को डाउन रेगुलेट करना है so that down regulating of a gene can be used by this particular phenomena, right? So again these are the certain examples you can just go through them. Okay, then in some woody plants self incompatibility stands as a major problem in fruit set and the breeding programs, and production of a transgenic apple trees they were able to self pollinate and develop fruit, so this can be done by this method again. Then fruit quality has also been assessed by silencing experiments. Several characteristics are involved. Transgenic apple fruits silencing key enzymes involved in autocatalytic ethylene production were significantly firmer and they had increased shelf life also. जबकि apple की shelf life वैसे भी बहुत ज़्यादा है. Then apples containing reduced amount of malodiven allergens were also obtained by expression of intron splice hairpin RNA content, right? So we can manipulate them. We can do the down regulating of certain gene and overexpression of certain other genes, right? So uh, it is possible to produce transgenic hypoallergic apples using RNA sources, right? So in citrus, the down regulation of putative thermostable packing methylesterases is projected as a possible solution for undesirable separation of a juice into clear serum and particulate phase. So in this RNA can also be used to achieve this goal. So there is a number of ways by which this technology has been used and has been exploited in the crop improvement. So this is all that I wanted to talk about the bean challenge. Though bean challenging is a very deep subject, the more you read about it, the more you feel that it is, right? So even when we cross protection, I last time I discussed the cross protection, right? जब हमने virology का पढ़ा था management में, so क्रॉस प्रोटेक्शन में हमने क्या किया था माइल्ड स्ट्रेन यूज किया था राइट टू प्रिवेंट द सिवियर स्ट्रेन तो इसका जो बेसिक बाद में पीपल व्हेन दे स्टडीड दिस मोर इन डिटेल दे फाउंड दैट उस फिनोमेना के पीछे भी जीन साइलेंसिंग और आरएनए suppression was the phenomena which was working there, right? So later it was found out by the people that this was the basic reason for that why they were not able to, or when the mice were there, the when you infect with the virulent strain, the why the disease does not take place. So the number of answers were basically explained by this particular phenomena, which was discussed or which was identified much later than the cross protection, though it has been used very widely since ages. Cross protection is not a new technique that we are using, right? Okay, so that's all from my side. If there's anything or any question you want to ask, this is open for discussion. Just go through. I don't want you to go very deep into this particular thing, but then I wanted कि आपको कोई अगर आपको gene silencing के बारे में पूछे या you should know the basic about it, that how it can be used in crop protection. But if the discussing the mechanisms and phenomena, that's a lot at this stage. Okay, so that's all from my side, I guess. We are left with a very small topic in this. I guess the another important topic that I would like to take up after this next topic would be yeah, edible vaccines. So this is again an interesting one. So in the next class, we can discuss about the edible vaccines and also the production of secondary metabolized by tissue culture. So how edible vaccines are being, this is vaccines. Vaccines are important. We all know that vaccines are very important to treat the viral infections, right? But there are a lot of difficulties in traditional vaccine systems which can be overcome if there's a plant derived vaccine technology that can be used. So this is this one paper which is edible vaccines and advancement in oral immunization. So this is 2017 paper, very good paper. I'll publish this paper also, and I'm going to discuss this paper only in the next class. That what are how they do it and what are the basic requirements for these kind of a studies. So very interesting thing. This is also so we are going to pick up in the next class. So we will wind up the class here, right? Okay, so please for attendance, switch on your cameras. Adhish Mishra.
Present, ma'am.
Ayushi Jain.
Present, ma'am.
Sam.
Present, ma'am.
And Devanshi.
Present, ma'am.
Ishita. Janvi.
Present, ma'am.
Kshitij. Kshitij. Okay, Manaswari.
Present, ma'am.
Amrita.
Present, ma'am.
Nehal. Rupal. Sahiba.
Present, ma'am.
Sana.
Present, ma'am.
Shivani.
Present,
Kanvi.
present,
Kriti.
ma'am.
Present, ma'am.
Kriti. Yeah, there. Yukta Yuvraj.
Present, ma'am.
Present, ma'am.
Okay, just a moment. I just don't go. I just want to ask. Uh, just give me 1 second. I'm just trying to open the attendance register. Okay, okay, fine. We can just do something. Yes, Devanshi.
Ma'am.
Ma'am, share,
Ma'am,
can
Devanshi.
you share your this week timetable with me? Actually, we have to like biostatistics sir have to publish some extra class, so it
Achha, so
won't
you
get.
want my timetable?
Yes, ma'am.
Yeah,
And
I'll
I guess yes, ma'am.
uh,
Ma'am, you have to also publish some extra classes.
yeah, I need lot of because plant pathology is 40 is like 67 I could take and we are still in 40s, right? Tissue culture, maybe few will do, two each week will be able to do, but in plant pathology, I definitely need more classes, right? I'll let you
Ma'am,
know.
then please, yes, ma'am, please share your free slots with me.
Yeah, sure, no problem. Yes, there was some hand raised. I don't know
Ma'am,
who.
ma'am, Shani this side. Actually, I wasn't able to unmute myself earlier. I'm present.
Okay, fine, no problem. Okay, bye bye. I'll see you tomorrow then. Thanks.
Thank you, ma'am. Excuse me, ma'am.
Yes, Sanjay. तन्वी तुम कुछ कह रहे थे?
Ma'am, can we present our PPT tomorrow?
हाँ, आपने कहा था ना tomorrow.
मैम मैम हमने आज बोला था, पर आज एमबीटी की प्लांट पैथोलॉजी की कोई क्लास पब्लिश।
आप अगर आप पब्लिश करवा दोगे तो मैं आज भी ले लूंगी. लंच में मैं बिजी हूँ और उसके बाद अगर फ्री हो तो करवा दो.
मैम वो तो दिवांशी कराएगी.
हाँ, तो you will have to talk to Devanshi, right?
मैम
अगर अगर
दिवांशी
ना कल कल का रुको. Let me see tomorrow what is tomorrow. Just a moment. Tomorrow is Tuesday. Okay, tomorrow we have class 12:15. तुम ऐसा करना 12:15 से 1:15 बजे तक है तो 1:15 से 2 भी पब्लिश करा देना. ठीक है. हाँ दिवांशी, मैं बात कर लूंगी.
थैंक यू मैम.